So good evening everyone, this is your 12th week session. So in this session we will be looking into structure based drug discovery, in which we will look into the structure based drug discovery and rational drug discovery. Then we will see the docking based virtual screening methods, how it has processed throughout the years, what are the challenges which are associated with it, and what is the future perspective on docking based virtual screening. Then we will see that what makes the small molecule an ideal drug, in which we will see what is Lipinski rule of 5, in which we will see what are the ADMET properties and what is structure based drug discovery, and we will see few case studies related to it, and then we will conclude it. And then later on we will practice few MCQs and we'll see the current literature, and then I'll take your doubts. So if we talk about the drug discovery, then it is a process in which we identify new candidate medications. So we create small changes in the chemistry, or you can say small changes in the structure of the molecules which are already known, to enhance their biological properties so that they can act as new drug candidates. So there are different stages of this drug discovery process. So the first step is target selection. So target selection means the protein on which you want to target a drug. So that particular step is known as target selection. That means you identify a biological target against which you want to design the respective drug. Second step is lead discovery. So it means you screen for compounds which will act on this target, which will bind to this target. So that comes under lead discovery, in which you will be, in the end, you will be left with a compound which has a suitable activity against this target. Third step is medicinal chemistry, in which after identifying the lead compound, that is your drug, you look for its optimization so that it has a better activity, it is most potent, and it should be selective towards that particular target. So all those optimizations comes under medicinal chemistry. Then we will perform in vitro studies. So this is the fourth step, in which we perform lab experiments to test that whether this drug is effective or not. So generally we test it on the cell-based systems. Then comes in vivo studies, in which we test it on animal models. So there are different stages in which we check that whether the drug which we have selected, it has either certain side effects. So this comes under various clinical trial stages. So now let's see the overview of drug discovery process. So the first step is target selection. For example, if somebody is suffering from a disease, then we'll find out that what are the proteins responsible for that disease. So that will be our target selection, in which we identify the protein which is responsible for the disease or the enzyme which is responsible. Then we'll find the compounds or the lead compounds which will bind to this protein so that we can inhibit its action, or we can enhance its action depending on that what role we want to see from that particular target. So for that we'll find out respective lead, then we will optimize it so that it can perform better, its activity is enhanced. Then we'll perform in vitro studies, various biochemical assays are performed, such as its activity is checked and all other criteria are checked. Then animal models are taken and then experiments are performed, such that we can see that whether this drug, although it is performing its reaction in vitro, but either in organism also it is able to carry out its activity or not. So for that reason we go for the in vivo studies, so in which we develop a model, then we validate that particular target protein, then we check that whether the drug which I have designed, either it is means binding to that particular target protein, or either there are some off target bindings, because we want that our drug should bind to that particular target protein such that it can inhibit its action. If it will bind to some other proteins, then it can disturb the entire biochemical reactions inside a particular organism. And then we manage the data based on that whether my lead compound or the drug is performing better actions, then I'll maintain the data related to that particular drug, so that in the later future we can design new drugs based on those properties. So one by one in the later slides we'll see in depth about all these properties. So the clinical trials are the stages in which we check that if somebody takes that drug, then what are the future actions or reactions that are happening. So each drug has to pass through these clinical trials to get approved. So preclinical means where we are performing these experiments in model organisms. Then in phase 1 we give it to the human organism, so in which we'll test it on human. So 20 to 80 participants are selected. So generally these clinical trials are majorly performed on the diseased people and not on the ordinary or healthy people. So on firstly at phase 1 we'll check it on the human by taking 20 to 80 participants. In phase 2 we'll test it on 100 to 300 participants, and in phase 3 we'll increase the number of participants. So the drug which passes through phase 1, that will be tested at the phase 2, and then ultimately it will be tested on at phase 3 level. And ultimately the drug which will pass through all these phases, that will be checked by the FDA. They will approve, they will check that whether this drug is safe, it is effective or not, either it has certain side effects or not. So after passing through all these phases, in the phase 4 it will be tested on 1000 plus participants, and then ultimately this drugs get approved. So this clinical trial is just to find out that whether the drug is safe in long run or either it has some side effects. So now we'll see that what are those small chemical changes which we brings such that we can change or we can bring about the large biological effect on that particular drug molecule. So there are certain compounds which are known as nucleoside analogues. So as the name suggests, nucleoside analogues, that means the compounds which resembles or which appears structurally similar to the nucleosides. And we all know that in a DNA, RNA, all these biomolecules, it contains nucleoside. So nucleoside is a sugar molecule along with the nitrogenous base. So that is known as nucleoside. So these analogues are the compounds which mimic these natural nucleosides but with identical hydrogen bonding properties. That means, as we all know, that adenine base pairs with thymine, so it shows double bond in DNA, whereas in RNA we see adenine double bondedly bind to the uracil. Similarly, guanine binds to cytosine with triple bond. So what these analog does is they resemble these nucleosides, and they will also show the similar kind of hydrogen binding abilities. So we'll see what are those nucleoside analogues. So the mechanism is that once they are incorporated into the DNA, they will block the chain synthesis and ultimately lead to the death of the cell. And the purpose is that it is used as a drugs to exploit the small chemical changes, for example OH modifications, to disrupt the DNA replication. So before looking into the mechanism and why it is needed, we will see what are the nucleoside analogues. So the example is this ribavirin, which is used in the case of hepatitis C. So in these disease we use these nucleoside analogues, and this is the nucleoside analog of guanosine nucleoside analog. So it resembles the structure of a guanosine. So whenever it will be present, so in place of guanosine, this particular analog will switch its position and it will not allow the DNA chain to replicate. Then comes adifovir, which is another nucleoside analog of adenosine monophosphate. So this also resembles adenosine. So in place of adenosine, this particular analog will bind, and then it will also inhibit the elongation of DNA chain. Similarly, another nucleoside analog can be produced as a result of substitution or certain additions. So these are simply the sugar moiety and the nitrogenous base, but there are certain changes which give rise to these nucleoside analogues. So similarly in this case, we have this trinitrogen which is attached to this sugar moiety. So this is the thymidine synthetic analog, and these two analogues which are represented in the bottom, these are the synthetic analogues, whereas the upper ones are the natural analogues which are naturally found in certain organisms or which are naturally observed, whereas these two are the nucleoside analogues which are synthesized in the lab. So we can also modify the sugar core. So in this case sulfur is attached, so this acts or this appears as a cytidine synthetic analog. So in place of cytidine, this particular analog will bind, because during replication, as we know that DNA polymerase attaches the complementary nitrogenous base or complementary nucleotide towards the extending or in the daughter DNA strand, so it will get confused. So in place of cytidine, it will select this particular cytidine synthetic analog, so which will ultimately leads to the termination of DNA replication. So these are the examples, and what are the modifications which are taking place is the removal of hydroxyl group. So from these nucleosides, there is removal of hydroxyl group which will give rise to these nucleoside analogues. or there could be use of L nucleoside analogues or incorporation of open chain sugar molecules. So as we can see in this case, it is an open chain sugar molecule in which the sugar, so this one is the sugar, so in this case it is like an open chain. So these are some of the chemical modifications. These are the small modifications, but still they can bring about the larger effect. Now we'll see what kind of effect. So for example, if some virus attacks our body, so in such cases these nucleoside analogues are used. So what they will do, as they resemble the nucleoside structure, so in place of that particular nucleoside, these nucleoside analogues will fit, and now they will not allow this particular DNA chain to elongate, because normally we require the OH group at 3' position of this sugar so that it can extend, it can form a phosphodiester bond with the next nucleotide. So in this case, because this 3' OH group is missing, as we know that removal of hydroxyl group can also give rise to nucleoside analog. So in this case, that hydroxyl group is missing, so now it cannot extend further, DNA chain cannot extend, so it will interfere with the replication of that particular virus. So these are some of the nucleoside analogues which are used in case of these particular disease such as hepatitis C, hepatitis B, similarly HIV and hepatitis B. So this is their role, this is the mechanism, how they work. So ultimately we can say that a small change in the chemical structure of a nucleoside can greatly affect the biology by halting the DNA synthesis and inducing the cell death. Because if DNA replication will stop, then the other machinery such as transcription, translation and all those things, they will also ultimately stop. So in this way, this highlights that how the small change can bring about the larger biological effect. Now we'll see how to find the lead compound, because firstly we know that in the drug discovery process, first step is to identify the target and next step is to identify the lead compound. So the lead compound is simply the drug which will bind to the particular respective target. So lead compound is a small molecule which is used as a starting point for further optimization. So generally in drug discovery, there are multiple steps of drug discovery process, and there are different types of drug discovery. So one type is knowledge based, in which we already know the drug. Now we are optimizing it to improve its activity. So in such case, this lead compound, it is already known, but we try to optimize it as more as possible. So similarly, they are saying that this lead compound, it is a small molecule and it is used as a starting point from where we are starting, and then we are optimizing it. So it guides the development of related small molecules. So in this, we are not designing some new drugs, but we are designing the related small molecules with similar structure, but now because we are optimizing it, so its biological activity will be improved. So the importance of these lead compound is that it is essential for the drug discovery and development, and these lead compounds, they are generally refined so that we can increase the efficacy of that particular drug, its selectivity and pharmacokinetics. So this efficacy means that it should be effective. Selectivity means that in some cases certain drugs can bind to certain off targets. So in that case, that particular drug will have certain side effects because it will also interfere with the other normal functional proteins. So in that case, during optimization, we make sure that now the lead compound should be very selective towards that particular target against which we are designing it. And pharmacokinetics means whenever the drug is in our body, then how much time it takes to get solubilized, absorbed, then distribute into various parts. So we try to add certain groups or some chemical groups such that it can increase the pharmacokinetic properties of that particular lead compound. So it comes under the why we need lead compounds and what are the things which we can do. So the databases which are generally used, so there are certain organizations which maintain the record of these small chemical compounds, and these databases, they can be categorized as publicly accessible such as PubChem, DrugBank, ChemSpider. So these are the databases in which already known drugs are present. So if you want to see certain structure, for example, if I want to design a drug against HIV, so in that case we can go into these databases and we can search that what are the drugs which are already known. Then we can study its structure, we can see where it binds, and then we can design certain new drug molecule by improving its properties. Then there are certain databases which are proprietary. So these are the databases which are maintained and restricted by certain organizations. So these are not freely accessible. You might have to pay or you should be working under them so that you can access these molecules, such as ACS, which is American Chemical Society, and it contains about 10 million compounds. Similarly, SciFinder, QSAR are some other examples of these particular databases. And then how to identify the lead? So it starts from the hits. So hits are the compounds with some measurable activity. So whenever you are trying to optimize the lead, so in these databases for each disease you will find multiple small molecules or multiple such small compounds, so that are referred to as hit, which has certain biological activity. Because our aim is to optimize it, so we will not call it as a lead. Previously it was a lead, but now because we are trying to optimize it, so for us all those chemical compounds will be hit. They have certain biological activity. So in this case they have the measurable activity. So starting with 10 micromolar, so this is the concentration which is required to bring about particular biological activity. So with this we'll filter out each and every compound and we'll make certain changes. So with this, each step by step we'll move to the lead. So lead will be the one which will have the lowest concentration, which is more effective, so which is less than 10 nanomolars, so which shows that its activity is improving and less concentration is required to bring about that activity. So we are optimizing the hits. So this is known as, this comes under the lead identification process, in which we are trying to improve the activity such that lower concentration dose is sufficient to bring about the larger effect. And this is the spectrum in which the activity is moving from the low activity to high activity by bringing about the changes. So this is how we find the lead compound. So if overall we have to mention about the drug discovery process, then we can say there are two ways in which the drugs can be discovered. So first one is lead driven approach, in which you already know that this particular drug is responsible for that particular disease. So from that database you refine it, you optimize it. So that comes under lead driven approach, in which you know the lead. Second approach is target driven approach, in which you know the protein, and against that particular protein you look for the binding sites, and then you find out that what small chemical compounds can bind to that particular site and inhibits its action. So that comes under target driven approach, in which you know about the target protein. And then target focused biological assays, in which you find out that what could be the optimum chemical compound which can bind to my target. So this will take us to the hit identification. So hit means all the chemical compounds that can bind to the target molecule which are not yet optimized. So this could be natural products, and in which we can use high throughput methods by look for the binding affinities, all those calculations can be done. Then lead identification. So from these hit compounds we'll identify the lead, in which we'll see that which has the highest activity, because hit can contain large millions of compounds. Then we reduce it, we shortlist certain compounds which has sufficient activity even with the lower doses. So that comes under lead identification. Now as soon as we have the lead, we try to optimize it further. We try to make certain small changes by adding certain hydroxyl groups or other groups to make it more active or make it better. We'll screen it. So this process is like a cyclic process, in which we keep on refining it, optimizing it, until we do not get stuck somewhere, we do not get saturated with all such changes. So this takes us to the lead optimization step. So ultimately this step is completed. Then comes preclinical development, in which we will now test these lead compounds. So we check for the pharmacology, in which we see that either the drug which we have further refined, either it has proper absorption, proper distribution, it can be metabolized, or it can be excreted out properly, whether it has toxicology or not, because it should not be toxic to the human body. So all these biochemical assays are performed in the preclinical development step, and then clinical trials are performed on the animal models, and then ultimately the drug which will pass through all these stages will be approved and it will be launched in the market. So this drug discovery process, it is quite time consuming, it's not that short. And this is the overview of how the lead compound is identified. So journal screening is done. So we have lots of small molecules which comes under the hits. And then in targeted library, we know the structure of that particular protein against which we want to design the hit. So this is also the similar overview as well. So firstly, primary assays will be performed, in which we check for the potency, that either the small molecule, it is potent enough or not. And then we perform secondary assays. So this considers the biochemical assays such as selectivity, solubility, stability, the inhibition coefficients, activation coefficients. So all these analyses are performed. And then actives are the compounds which shows better of these activities. So they comes under actives, which are active, which can bind to a selective protein target. So the criteria is it should be potent, selective, it should obey the ADMET properties. So in the later slides we'll see the Lipinski rule, in which we'll see that what are the criteria which a drug must follow to be efficient or efficacious. Its efficacy should be high, so that comes under the ADMET properties. And then patentability, and ultimately lead molecules are identified and followed by the clinical trials. So there are multiple steps which are required to optimize the lead compounds, so that also we'll check. Okay, so now we'll begin with the traditional drug discovery process, and then we'll move to the rational drug discovery. So this is just the starting point, that how that started. So traditional drug discovery, it is a process in which we identify the drugs from natural sources, or this also includes the accidental findings. That means you do not have you're not trying to find out something, but accidentally you find out certain drugs. So accidental findings through random screening or trial and error methods. So the first process under traditional drug discovery is random screening. So in this, you randomly screen synthetic compounds or chemicals or natural products using experimental assay methods. So in this, you do not know whether that compound is effective against certain target or not. You randomly take certain compounds and you are just exploring it. So this also kind of exploratory studies. So two major approaches in random screening are targeted screening of compound classes, such as you are specifically targeting certain classes of compounds such as alkaloids, flavonoids or synthetic libraries. And then comes random screening. That means any medicinal plant which is known, you simply select those plants and you are just trying to find out whether it has some relevance. So here you can see that some flowers or some medicinal plants are taken. So it contains nearly 5,000 to 10,000 compounds. So we are not taking directly the plants, we are talking about the chemicals or the molecules which are present inside these plants. So it could be around 5,000 to 10,000 molecules or compounds that are taken, and then we screen them. So it takes about 3 to 5 years. In the screening, we'll see their structures, we'll see that to which protein targets they can bind, what are the properties of these small molecules. So that takes around 3 to 5 years. Then we'll perform preclinical stages, preclinical steps, in which we'll check for the activity of these compounds. So out of all these compounds, approximately 250 could get filtered out, other might not have that sufficient activity. Then clinical trials are performed in vivo on animal models. Then only 5 compounds could pass through it, and ultimately after checking for the ADMET properties, all these properties and regulatory approvals, it can take up to 1 to 2 years. And the compounds which can pass through this, they will be available at the market. So that takes us to one new drug. So that means we start with lot of compounds and we are left with just one new drug. So these numbers are not fixed numbers, this is just an estimate that how at each step the number of compounds keep on decreasing, because we are trying to improve the activity and all these things are filtered out. So this is the random screening. Next thing is trial and error method. So this is also based on trial and error. In this you do not have any knowledge, pre-existing knowledge about the drug, but you are just trying it, such as you can take the compounds from berries, roots, leaves or barks for medicinal purpose. So we know that in India, the ancient literature which is present, we have a lot of medicinal plants which are already mentioned in scriptures and all those things. So from there we can select certain roots, leaves or barks, and from them we can isolate the chemical compounds, which we can extract chemical compounds which are present in them, and then we'll perform biochemical assays and we'll check that whether they have, what are the structural properties these compounds have and to which targets they can bind to, and then same stages the compounds will pass through, and then ultimately the compound which has better potency, which is effective, that will be approved, and ultimately the drug will be out in the market. So the example is of cinchona bark, which contains quinine, which is used to reduce fever in malaria. So this knowledge was previously present in some scriptures, so that's why this bark was taken and the compound which was isolated from it was quinine. So this shows you that this trial and error method can also be used to design the drugs against particular disease. So this is based on the ancient knowledge about these particular medicinal plants. And second is of neem leaves. So it is used for the anti-inflammatory and antibacterial purposes. So the research started from these leaves and the chemicals were isolated and then checked that what is the chemical which is present in these leaves which is responsible for this activity. So this is about the trial and error method. Next approach is ethnopharmacology approach. So ethno, as the name suggests, ethno, that means it is belonging to certain cultural groups. So in this we study those medicinal plants which are specifically used by certain cultural groups, and it involves observation, description and experimental investigation of indigenous drugs. So if certain cultural group is using certain plant and then they have seen certain effect of using that plant, then we proceed with that particular plant. So example is of Andrographis paniculata, which is used for dysentery, which has some active compound, and that active compound was identified as andrographolide. So in this we start with the plant which is already used by that particular cultural group. We isolate the chemicals which are present inside that plant, for which we will perform different assays. So different biochemical assays are performed, such as DNA fingerprinting, immunoassays, chemometrics, and then that particular plant, its biological study is also done, such as its taxonomy is identified, its macroscopy, microscopy is done, its chromatography is done to know that which are the different chemical compounds present in it and their properties. Then spectroscopic methods could also be used to authenticate the small compounds which are present in these plants. And then the extract which is obtained, its biological and toxicological screening could be done to find out that whether it has some chemical effect or whether it is toxic or non-toxic. And isolated pure compounds are further used for the molecular modeling and in silico screening. So in these molecular modeling, generally it is observed that whether this compound is stable, what is the structure of this compound, and then structural modifications are done to develop the new analogues of that particular compound, so that we can increase the database of those chemical compounds, and we can find out that either some of them could be better lead compounds. So this is like we obtained a hit and we are optimizing it. So in this case, optimization is done by using this molecular modeling and making certain structural modifications. And ultimately in vivo studies are done in which it is identified that whether it would have the effect in the live organism or not. Because in certain conditions the drugs appear to be effective in vitro conditions, but as soon as we give them in vivo, then because the conditions are quite different, so in that case the drug might not behave in the same way. So that's why this in vivo screening is very important. Yeah, so this is also one of the flow chart which shows that ethnobotany or ethnopharmacology, the first step is the plant selection, and then we authenticate that plant, we extract chemical compounds, and for that fractionation and isolation is done. So pure compound is obtained. Now its structural elucidation could be done so that we can synthesize it, and we can also synthesize some new chemical compounds related to that particular structure. So other thing could be molecular modeling or in silico screening, in which we can modify that particular structural compounds and design some new analogues. And for that particular compound we can do the biological screening of that compound, and we can check that either it is toxic or not. So ultimately the compound which passed through all these tests will be the effective drug molecule. Yeah, so this comes under the traditional approach, in which we start with a particular plant or we start with drug molecule, and then ultimately we perform different experiments and that particular drug molecule is out. But then there is another approach, which is the modern approach, which is computational chemistry approach, in which we have the database of multiple such lead compounds, and we are just optimizing them to improve the activity. So if we start from knowledge base where the lead compound is already known and then we are optimizing it, then it comes under the modern approach. So So the fourth type of traditional drug discovery method is classical pharmacology, or it is also known as function-based approach. So in this, drug discovery without prior knowledge of drug target. So in this, we do not know that what is the drug target which that particular drug is targeting. It is just based on measuring the complex biological responses. For example, certain drug, it is better for the seizure prevention, or it is reducing the blood sugar. So we do not know which protein it binds to, how it is, what is the mechanism behind it, but we know that this particular drug or this particular lead compound, this is bringing the functional effect. So this type of study comes under the classical pharmacology, in which we know the function but we do not know the target. For example, the foxglove plant, it led to the discovery of digoxin, which is a cardiac glycoside. So this is generally a blood thinner which thins the blood and which can help the patients who have the clots in the blood or who could suffer from the cardiac arrest. So this was known, but how it works was not known. So this comes under the function-based approach, or this is also known as forward pharmacology, where we know the function of the drug but we do not know where it binds to. And then the reverse pharmacology is the opposite of it, where we know the target protein to which we want to bring effect, and then we go for the discovery of the lead compound. So CHA's clinical observations of behavioral effect of non-psychiatric drug. So we start with the effect, and then we understand that what is the mechanism, so which comes under the reverse pharmacology, where we are looking for the mechanism of the drug, and then synthesis and testing of the novel drug in animal models of behavior. So after this observation is done, after the mechanism is identified, now we can synthesize the novel drug by bringing about modification in those drugs from where we started. So this is simply the optimization and ultimately target validation in the patients. So in vivo studies are done in which the target is identified, and this is also the another flowchart of classical approach versus the reverse pharmacology. So this is also the same which I have mentioned, that in classical approach we start with the function, in which we know that this particular drug is bringing about this function, and then we start from here and we ultimately identify the mechanism. Mechanism means we identify the molecular target on which it acts and how it is bringing that effect. In reverse pharmacology, the approach is opposite, in which we know the target protein and ultimately we are identifying different drug candidates, and in the end we are getting that drug candidate. So next type is serendipitous discoveries, which are identified by accident. So bacteria was identified in 1670s by Van Leeuwenhoek, which linked to disease established in 19th century. So till that time it was not known that these bacteria are also responsible in certain disease conditions. And discovery of antibiotics, penicillin, it came from the accidental findings. So penicillin, which is significantly responsible for this antibiotic, which is the most well-known antibiotic, which was discovered by accident. So it significantly reduced the wound related to pneumonia deaths in First World War, which reduced the numbers from 18% death rate to less than 1%. So then it was known that these bacteria are responsible for certain wounds, and these antibiotics, they can help to reduce the death rate. Yeah, so this was the Petri plate which is observed at that particular time. So this is the serendipitous discovery, in which this particular scientist have left the plate, and he observed, when he came after a few weeks, he observed that wherever the penicillin colony was growing, so in that case it inhibited the growth of bacterial colonies. So in this case there is no growth of bacterial colonies, whereas the colonies which are away from this penicillin colony, they are growing well. So this shows that penicillin is having some kind of antibiotic properties. And then another example is of the sweet clover plant. So in this case, this is the historical discovery of this drug, which is the dicomural drug, which is a blood thinner. So in this, kettle was fed on the spoiled sweet clover, due to which the kettle, it developed unexplained bleeding. So Carl Paul Link, he was the scientist who investigated it, that why this kettle is showing the bleeding. So he observed that there is some compound which is present in the blood of kettle, which was dicomural, which is having the anticoagulant response. So this was not allowing the blood to get coagulated, due to which there was excessive bleeding. So this was a serendipitous discovery in which this was not decided. So ultimately it was known that that kettle was fed on the sweet clover plant. So it was known that this particular compound is coming from the sweet clover plant. So from where it was known that what is the source of this particular drug or lead molecule. So this is also the serendipitous discovery, and this is beneficial as well because this is the blood thinner. So now there are few challenges in drug discovery. So first challenge is high cost and time investment. So this is quite costly process and also taking a lot of time because you are trying to optimize it. So this is time consuming. So about 70% of funds, they are lost due to failure in clinical trials, because when you are optimizing lead, all those things you are doing, it is also taking a lot of time, and ultimately when you are passing through different clinical trial stages, and if your drug fails in that, then you again have to start from zero. So which shows that it is taking a lot of time, and this is not very promising. You're not sure that the compound which you are proceeding with, it will be passed or not. It might get failed. And then the next problem is high developmental costs, because as it is taking a lot of time, so now the drug which will be approved, that will become very costly. So if it is very costly, then people will not be able to afford it, and there will be market competition because your competitors will also be selling the drug. So due to which you need to reduce the price. But if you reduce the price, then you have to deal with the losses. So these are few of the challenges which are associated with drug discovery process. So because in this case we are performing the experiments, means we are performing wet lab experiments. So now we need to bring about certain changes so that we can reduce the cost, and we can also reduce the time which is taken from the drug discovery process. So then comes the another approach, which is rational drug discovery. So previously it was traditional drug discovery, in which we are experimentally testing everything. But now in rational drug discovery, this is also divided into two main approaches. But in this case, we are using the different computational approaches. So we are shortlisting, or we are performing in silico studies first, and then the promised or the most effective hits are only selected for further experimental investigations. This will reduce the time as well as the price or the cost which is associated with all those experiments. So the rational drug discovery process, it can be classified into two main approaches. First one is ligand based discovery, and next is structure based drug discovery. So this is also same as that of lead based versus target based. So this is same as that only. In ligand based drug discovery, it is used when we do not know the structure of the target molecule or the target protein on which we want to target a drug. And this method utilizes the geometric rules or physicochemical properties. So in this we already know about the ligand. So ligand is a small molecule, or you can call it as a drug, which will be a future drug. So that particular compound which can bind to the protein. So in this case you know the information of the ligand, but you do not know to which protein it binds. So you can take help of the physicochemical properties so that you can design new such lead compounds, and you can ultimately proceed. So this uses geometric rules, in which we look for the structural properties and physicochemical properties. So the key concept is pharmacophore models derived from QSAR, which is the quantitative structure activity relationship studies. So pharmacophore means where you study the structural properties of some small molecule, and then you relate them with how the structure is related with the activity. So in this way you make changes in the structure, and then you correlate it with the activity, that whether the activity will go up or it will go down. So in this case we are trying to predict that which structure will have what activity. So this is the key concept of ligand based drug discovery, and the purpose is to screen the compound libraries based on the ligand features. So instead of making every single change and then experimentally testing it, we are performing it in the in silico conditions or in computer, because we have already available data set in which we already have the chemical databases such as PubChem, Drug Bank. So we know all the chemical compounds and their respective activities. So from there we take that information, we train our data, and then we develop new compound, and we predict that what could be the possible activity which is possible for that compound, so that we can only proceed with those compounds which show better activity in silico experiments. So this will reduce the cost as well as time. So don't worry, in later slides we'll see in detail, so don't worry. And then second approach is structure based drug discovery. So this is used when you know the structure of the protein which you want to target. So the method is molecular interaction calculations. So now as you know the protein which you want to target, so you will see that what are the binding sites which are present in that protein, what are the groups which are lying in the binding site, so that to those respective groups you can design the drug molecule. So test molecules and target are depending on the molecular interaction calculations. The key concept is that you use the scoring functions. So you will design certain chemical compounds, or you can take those compounds from already available libraries, and then you will see that what is the binding affinity of all those compounds. So the compound with better binding affinity will be selected as the lead to proceed with further other tests. So you use scoring functions to calculate this binding affinities. So requirement is that you must require the 3D structure of the target. So if you know the 3D structure of the target, then you can proceed with the structure based drug discovery. And this is what it shows. So homology modeling, homology modeling is a method in which you try to predict the structure of the protein which is unknown or whose structure is not yet solved. So for that we can go for homology modeling. And if it is solved, that X-ray crystallography, NMR, cryo-EM, all those methods. So if the structure is already solved, then that also comes here. And then non-actives means these are the lead compounds, or you can call them as ligands. So these are the ligands or the possible drug candidates. So they are taken, they are pooled. So this will be a hit compounds pool. So these are hits, which are the known actives. And from them you will further refine, you will further optimize the lead compound, and ultimately you are obtaining the lead. Then later on other in vitro, in vivo studies are performed, and then the drug will be out in the market. So the same steps will be here only. So then ligand based drug discovery. So we start with the ligand based pharmacophore, in which we look for the structure of the ligand. So ligand are from these known bioactives or known actives. These are the ligands or the drug molecules. So we study their structure. We use 3D QSAR, in which we look for the 3D properties of this particular structures, and we see how the structure is associated with the activity, which structure shows what activity. So those models are developed. Then after developing those models, we look for the 2D similarity based search, in which we design multiple chemical compounds and we'll see how they are similar to one another, what are the similarity in them. Then scaffold hopping. So these are some of the methods in which you generally optimize the structure so that it can give better properties. So then ADMET predictions are done. For all those compounds, we predict the absorption, distribution, metabolism, excretion, toxicology. So these are multiple parameters which are studied to check that whether this particular drug is effective or not. Because if it is getting absorbed but it is not getting properly distributed throughout the body, then we cannot consider that as effective drug. Or if it is not getting metabolized, or if it is not getting properly excreted, or if it is directly getting excreted. So all those studies need to be done to make sure that the drug, it is properly binding to the respective target or not. So this all comes under ligand based drug discovery. On the other hand, in structure based drug discovery, you know the target protein. So you try to predict the druggability. Predictions are done, in which you see the structure of that particular target, you find out its binding sites, and then respective to those binding sites you use different chemical compounds and you perform the docking. So in this, docking based virtual screening is done. So docking means in the binding pocket of the protein, when you look for the binding of the ligand molecules or the small molecules, you are looking for its binding in the binding pocket of the protein. So that is docking. That means you are docking that particular small molecule in the bigger protein to check whether with what affinity it is binding. And then structure based pharmacophore is done, in which you look for the structure of the lead compound, you optimize it further, you perform molecular dynamic simulations, because in molecular dynamic simulation you will see that what is the energy, which structure is most stable. So those studies are done, and ultimately de novo design could be done, in which you can design the drug from scratch. So in de novo design, because you know that the target protein is present and what are the binding groups which are responsible for binding, so from there you can design the drug by keeping those functional group and adding the scaffold or adding the backbone to it. so which comes under de novo design. So all these main topics we will see in the later slides. So this is about the rational drug discovery, and this is also known as computer aided drug design, because in this computer is generally used for the screening. And similarly, structure based drug design versus ligand based drug design, and these are all the steps which are generally used. And you can also use machine learning approach to speed up the process, because from the quantitative structure activity relationships you know that which structure will perform or will give which activity. So in that way machine learning models could be developed which can quickly screen millions of compounds in the library. So this will help to reduce the time and increase the speed as well. Yeah, and if you want to study it in details, and these are some of the points, the advantages versus limitations of each of these approach, so you can go through this. So now there is one of the example of rational drug discovery. So the drug is cimetidine, which is ADMET is the name of that particular medicine. So the problem was that problem identification first of all. So biological target, so histamine, it triggers the release of the stomach acid. So our goal was to develop a histamine antagonist which can be reduce the effect of histamine to prevent the acid secretion. So validated target is histamine receptor. That means you want to design the drug which can bind to histamine. So this is my target, and my ligand or my drug will be the one which will be binding to this. So this comes under the structure based drug discovery approach, in which I know the target of structure of my target. Now lead will be discovered. So how to discover the lead? So approach is to synthesize histamine analogs with systematic structural variations. So in this different variations are done in the histamine. So those variations, after the variations are done in histamine, they will be like analogs. They will mimic the structure of histamine. So they are referred to as histamine analogs. Now lead compound which was identified was guanyl histamine, which showed some of the antagonist properties to that of histamine. So now this is the step of lead discovery, in which the lead compound is identified. Now it is optimized further. So for optimization, its derivatives are improved. So from guanyl histamine certain modifications are done. Then burimamide was synthesized. So burimamide, this is the compound, and thiaburimide was also synthesized for better activity, so which have better activity. So there are certain changes which are bring about in this group where the sulfur is attached. So buramide compound was synthesized, which is the histamine analog and which has antagonist property to that of histamine. And then later on further optimization steps were done. So next optimization step was to make it more potent and orally active, so due to which other modifications, other hydrogen was attached, and then double bond S. So this was some modification was done in this particular group. So after which we obtained metiamide. So in metiamide the problem was that it was containing sulfur, so it was toxic in clinical trials. So now this sulfur group need to be replaced. So then another drug was developed which was cimetidine, in which this toxic group is replaced. So instead of this particular group, it is replaced with the another group which is this one. So now which resulted in an effective group, and this was well tolerated hydrogen receptor antagonist. So this is generally used, and then another successors also came, because with time different optimizations were further done. So this was later replaced with the rentidine, in which this group was further replaced with this particular group, so which has better safety profile than cimetidine. So this gives the overview of the approach, structure based approach, that how different modifications are done to optimize the lead compound which can act as the antagonist to the existing target protein. Now ligand based drug discovery. So as we know, in ligand based drug discovery we use the QSAR modeling, which is quantitative structure activity relationship modeling. And we have also seen that ligand based drug discovery means we have information of the ligand but we do not know the structure of target. So QSAR models, they are the regression or classification models which are used in chemical and biological sciences, and the main role of this QSAR model is to relate the chemical structure to the biological activity. Because we know the structure of my ligand, so we predict that what could be its activity based on the available information. So core principle is that it uses mathematical relationships between chemical structure and biological activity using physicochemical or derived properties as descriptors. So in later slides we'll see what are the descriptors. So if we want to see what is the mathematical relationship, how it identifies. So it assumes a mathematical function which is y, which is predicting the activity, which is the biological activity, and this is f represents the function of x. So x is that particular ligand, and function means how the activity is related to this particular chemical compound or structure of this chemical compound. So this function will tell you that how they are interlinked, so that for unknown x or for unknown some compound you can predict the y, that is biological activity. So this is the mathematical relationship. X is the chemical descriptors, which is a structure based parameter, so which has certain features which tells you that what is the structure, such as it could be hydrogen acceptor sites, hydrogen donor sites. So all those things comes under the chemical descriptors. Then data is taken for multiple compounds for which we know the activity values, because whenever we are doing any predictions or QSAR modeling, so in that case we require the data, we require knowledge. So that pre-existing knowledge, we design a classifier which can further classify based on that particular knowledge. So we start with multiple compounds for which we know the activity, and then we find out that what could be the formula or what could be the function, how the structure is linked to the activity. So after that particular function is known, which comes under the algorithm development, in which we use different statistical analysis approaches to create a function that can predict y, that is activity, from x, that is structural descriptors or structural features of the compound. So algorithm should output the values which should be close to real experimental data. So in this case we have two matrices, one is the molecular descriptor and another one is the response variable, which is the activity. So we find out that how they are linked. We perform statistical analysis. So from predicted values, because this will be my training set, and some of the compounds from this available data for which I know the activity, I will keep them as a test sample, so that I can verify or I can validate that with the prediction whether I am getting near or similar activity as that which is mentioned in the data set. So all those analyses are done, and ultimately if the analyses are right, that means I can proceed with that function. If they are not right, if they are not close enough, then we need to refine the function. So we'll not go into the detail of mathematical equations or calculations, but this is just overview that what is mainly done, how the QSAR studies are done. So model need to be validated by looking for the difference. So you might hear some term like partial square or least partial square. So all those things comes under the validation steps. So those procedures are done to know that how much the predicted values vary from the known values. So if they are more close to known values, that means our model is good. We can proceed with that QSAR model, and now we can give new compounds and we can predict the activity. So this step is generally to reduce the time so that we can shortlist the lead compounds, and ultimately for those shortlisted compounds we can perform experimental studies. So it is critical for ensuring the predictive accuracy and generalizability. So that's why this validation step is very important. Okay, now we'll see that what are the descriptors, because here we have seen that this X, it includes descriptors. So what structural information do we keep in X? So that is the descriptor information. So molecular descriptors, they describe the aspect of molecular structure or property. That gives you information that what is the structure and physicochemical properties. So these descriptors, they can be classified into different classes based on their dimensionality. So first one is 0 dimensions, so which includes molecular mass, atomic count and atomic mass. So for any compound, if we list these properties, then they come under 0D molecular descriptors. Then comes 1-dimensional molecular descriptors, which encodes substructural fragments, which tell you that what are the substructural fragments which are present, such as if benzene ring is present or if carboxyl group is present. So these are the substructures of bigger chemical structure. So this information is kept in the series of such strings or 0 and 1. So there is some way to represent. Each fragment will be represented with particular these binary digits or 0 1s. So this comes under 1D molecular descriptor. Then comes 2-dimensional molecular descriptors, which tells about the connectivity of atoms or molecular graphs, that how they are connected. So that information is under 2D molecular descriptors. Then comes 3D molecular descriptors, which tells you spatial orientations, and these 3D descriptors, they can be categorized into internal 3D descriptors versus external 3D descriptors. So internal means which are invariant to rotations or translations. So even if you bring about certain rotation in this 3-dimensional structure, so that is not considered in internal 3D descriptor. It considers the entire 3-dimensional structure to be rigid. It does not bring about any change during rotation or anything. Whereas in external 3D descriptors, this uses 3D coordinates with fixed frame. So if you change the frame, then the structure will get changed. So in that way, it is sensitive to the rotations and other translational movements which are done. So each frame will have each frame of this single particular protein can be different. Then comes different descriptor categories. So these are different dimensions in which the descriptors are present. Now these descriptors could be divided into different categories. So first is constitutional or information descriptors, which is simply giving you some kind of information such as the molecular weight, chemical elements which are present, carbon, hydrogen, nitrogen, sulfur, or hydrogen bonds or double bonds. So these informations comes under information descriptors. Then comes physicochemical descriptors, which will tell you about the physicochemical properties such as lipophilicity, polarizability. Then there are topological descriptors, which tells you about the atomic branching and connectivity. So this topological descriptors, they comes under this 2D category, in which it will tell you that either my structure is branched or it is unbranched. So there are different ways to perform these calculations or to identify these descriptors. So we will not go into the detail. So this comes under topological descriptor because this is telling you about the topology of particular chemical molecule. Then next are the electronic, geometrical and quantum chemical descriptors, which will give you the idea of electronic properties and its geometrical orientation. Then next one is fragmental or structural keys. So this 1D descriptors comes under this fragmental or structural keys, which gives you information about different fragments which are present in your structure. So based on it also we can identify or we can synthesize new drug candidates. So these are all the descriptor categories, and different tools which are used for calculating these descriptors are RT-Kit. So for example, if you have from drug bank or other database, you obtain certain chemical compounds. So now if you want to find out the descriptor properties, so you can simply use RT-Kit. You can provide those compounds and you can select which molecular descriptor property you want to see, and you will be able to get it, so that in later you can perform QSAR modeling and you can relate how the structure is linked to the activity, so that you can design new drug candidate by bringing about modification in those chemical compounds. Now there are certain assumptions in the QSAR modeling, such as the effect is attributed to model compound and not its metabolites. So this is one of the assumption. Next is that the proposed conformation is assumed to be the bioactive form. Next is all model compounds share the same binding site. Next assumption is bioactivity results from direct molecule target interaction. And fifth assumption is pharmacokinetics, solvent effects, diffusion and transport, they are not considered. So these are some of the assumptions which not always hold true in many cases, because in some cases the model compound, its metabolite could be effective, or it's not that only model compound is giving rise to certain effect. Maybe it get metabolized in the body and its metabolite is also bringing about certain changes. So all these things are not considered while doing QSAR modeling. Okay, so now let's start with this. So if we have known ligand structure, so this is ligand or ligand based drug discovery. So we know the ligand, then different tautomerization or protonation states. So these are the changes which are brought in the structure of the ligand, and then its conformation is searched. And similarly for protonation states also, we look for different conformational structures of that particular input ligand, and then descriptors are calculated for all those structures. Then average descriptor values by Boltzmann distribution is obtained. Then in this case we generally select that which descriptors better highlight or which shows better changes among different structural values. So for example, if I am looking for hydrogen bonding or number of hydrogen bond donor groups, and it is possible that two of my compounds which are different in the structure but they might have same number of hydrogen bond donors, so in that case that particular descriptor value is not sufficient to discriminate among those two structures. So generally what we need to do is we need to average the descriptor values. We need to find that particular descriptor which is able to distinguish uniquely the two chemical compounds. So that thing is done. Now QSAR modeling is done, in which how the activity is associated with the structure is done. So in this case activity is identified by using SPR curves which give binding affinity information and the kinetic information for these ligand molecules, that what are the binding affinities with the target compound, and different kinetical properties are also studied. After all these studies which have been done, or this could already be available in some literature, so from that also we can take it, and then we will perform the modeling in which we will train our model. So now we know that this particular structure has this activity, so that will act as my training set. We will use QSAR modeling steps. We will use those mathematical relation in which we identify the function which links the structure to the function or biological activity. So that particular function is derived, training is done, and then on test set we check that whether our model is good or not, and ultimately we will give new compounds and we will predict the activity from them. So this is the overview of QSAR, and this we already have discussed in previous slide as well. So this is just the revision, you can say. So in this way ultimately modeling is done. So this is how the QSAR model are developed and how we can predict the activity of unknown compounds. Then there is another descriptor which we have seen in the previous slide, which was the topological descriptors, which tells you about the topology of the structure of that particular compound. So there is the term called Wiener index. So now whenever we are performing these activity studies or different biochemical tests, so there is one index which is also quite helpful to find out the boiling point of alkanes, which is known as Wiener index. So according to chemical graph theory, this Wiener index, it is sum of the shortest paths between all pair of known hydrogen atoms. So in this we try to find out what is the shortest path. Shortest path means if we start from one atom, then to reach to other atom, then how much steps do we need to take, which means that if my structure is branched, or if branched means that different groups or different atoms are close to each other. If it is unbranched, that means the structural means different atoms, they are lying far away from each other. So in that case shortest path will be long, it will take a lot of time to reach from one atom to other atom, whereas in branching the path length will be small. So which means if my structure is more branched, then that means it will have greater Wiener index. So greater Wiener index means, in this as you can see in the graph, with increase in the Wiener index boiling point is also increasing, which means that if my alkanes, if there is branching, or if they have further so in simply it means that more the distance between different so it is the sum of the shortest paths between all the pair of known hydrogen bonding. So if the shortest paths are as it increases, the boiling point will also increase, which means that if the size of alkanes will increase, if there are more number of such atoms, then ultimately this is expected that if number of such known hydrogen atoms increase, then it will give rise to increase in the boiling point, because number of interactions will also increase. So this reflects the molecular topology and it correlates with the properties like boiling point. So this shows that how by knowing the topological descriptors we can even identify the boiling point among different chemical compounds, and in this example is taken for the alkanes. So this is to show you that how different descriptors can give you idea of different properties. Now another aspect is of 3D QSAR, so which is three-dimensional QSAR, and the model is receptor surface model. So hypothetical model is based on molecules 3D shape and activity data. Then we perform optimization, which involves atomic partial charges, surface fit, etc. So these are different changes which are brought about in the structure of the target protein to optimize it. And different descriptors which are used involve van der Waals energy, electrostatic energy, total non-bonded energy. So these are different energy calculations. For different structures, these calculations are done, that what is the overall energy of that particular protein structure, because this is based on the receptor surface model. So it uses GPLS, that is generalized or partial least square statistical method, for model development. So in this, all those steps are not repeated, but the steps will be same, but we are starting with the receptor. So receptor is like the target protein which is going to receive the drug molecule. So that particular target protein, when it is studied, when its three-dimensional structure is studied, then it comes under 3D QSAR, in which for that particular target protein we look for different energy calculations. So these descriptors will give you information of these targets, and ultimately we can predict or we can design the QSAR model respectively. Yeah, so this is the representation that if we have three-dimensional structure or coming from homology modeling or X-ray, so in that case we will predict the binding site in that particular target protein, and after identifying the binding site we will search for active compounds, or we can search for the ligand molecules which can possibly bind to my target protein. We can perform docking studies to look for the binding affinities of active compounds with my target protein. We will screen them based on scoring. We will only select those lead compounds which have better binding affinities to my target. So that will take us to compound optimization step, in which we are optimizing the lead compounds or the active compounds. Ultimately we will obtain the receptor image. We will perform pharmacophore modeling or pharmacophore mapping, in which we look for the structure of my receptor or target protein. I can also change the structure of my target protein to improve the binding to the active compound. So different optimizations can be done to ultimately move to the better drug target, or you can say to find the better lead compound. So active compounds will be identified and conformational search is done. So this is same as what we have discussed, but in this case we are starting it with the target. In previous we have started with the ligand. So depending on from where we are starting, the names could vary, but the overall workflow of QSAR is the same. We are just trying to link structure to the activity. Now, advantages of QSAR in predicting biological activity. So it predicts activity of many compounds without experimental data, because in QSAR you already take the help of existing knowledge. You already know what is the structure and what is the activity, so you design the function or you use mathematical relationships to identify that function, and then you are simply predicting it. So you are not performing experiments for the new compounds. So in that way it is helping you to perform such calculations without the need to perform without any need to perform the experiments. Second thing is, it helps you to identify the key molecular properties which are worth studying. So because in QSAR you are looking for the structural descriptors, so with those descriptors you will be able to know that what particular descriptor is more responsible for activity variations. So those properties you can further study. Then considered green chemistry, that means there is no chemical waste because it is in silico approach, so you are not wasting many chemicals. And it also reduces time and cost of in vivo and in vitro testing, because it will take less time because you are using in silico approaches. So in computationally you are testing so many compounds, so in that way it is reducing the time and cost. But there are certain disadvantages. So first disadvantage, that it lacks mechanistic insights into biological action. So as we have seen when we were looking into assumptions of QSAR, so in this case we do not know about the mechanism. Although at the level of structure we are able to predict the activity, but we do not know that when that compound goes in vivo or goes into animal model, then it will exist in that particular state, or either it will get metabolized. So all those mechanisms we do not know. So that is one of the disadvantage. Second is that it may lead to inaccurate predictions of pharmacological and biological activity, because it is based on function. And in biology or in chemistry we know that there are many exceptions which exist. So sometimes it's not that each molecule will follow that particular function which you have derived using QSAR. So in that case, certain predictions could be inaccurate as well. Yeah, so in this case, it is shown that steric bulk here is good, which is represented with this red. So this is showing different groups. So these are different type of chemical groups which are present in my target protein in structure or in the target protein. These are different chemical groups which are lying in the binding site, which are highlighted to simplify it. So in some cases, steric bulk is good, in which different groups lying close to each other and there is some steric clashes which are present, but it's helpful. Whereas in this case, this electrostatic coefficient or electrostatic groups are also present in my target compound or target binding site. Similarly, these other groups are also present. And then this is the another compound in which steric bulk is good, but in this case steric bulk here is good, and then high electrostatic variance is lots of change here. So in this case, this is simply showing you that two different compounds or their binding site can vary. In one place, the electrostatic interactions might be helpful, whereas in other compound, maybe electrostatic groups are missing, but still that compound is also the optimum compound. So this is to highlight that not all the descriptors will follow the same, means by looking at just few examples, you cannot know that which descriptor better links to activity. In some case, electrostatic groups can lead to increase in the biological activity, whereas in other compounds, those electrostatic groups might not bring any change in the activity, or they might reduce the activity. So it's challenging to decide which descriptors better links to the activity. So this is to represent that only. Yeah, then comes ligand based drug discovery, in which we have pharmacophore development steps. So pharmacophore means this is the description of molecular features which are essential for ligand recognition by biological macromolecules. So biological macromolecules means these are the targets, whereas my ligand is which is the future drug molecule. So ligand is the active compound which is getting attached to the target molecule or the macromolecule. So these models, they help to understand how structurally diverse ligands bind to common receptor site. So in pharmacophore, we look for the structure of different ligands and we see that how they vary in their binding to a common receptor or common target protein. So this is simply used for de novo drug design and virtual screening to identify new ligands for the same target. So in this could also be called as optimizing different leads or identifying novel drug candidates. So typical pharmacophore features are hydrophobic centroids, aromatic rings. So pharmacophore, we know that these are the structural properties. So what could be different structural properties? They could be hydrophobic centroids, or they could be aromatic rings, hydrogen bond donors, acceptors, because they are responsible to bring about certain activity. So these are the features which are mainly considered. And a robust model includes both hydrophobic volumes and hydrogen bond vectors. So mostly these are the groups which are responsible for different interactions. So they are mostly well studied pharmacophore features which are mainly considered. So in developmental process, firstly we identify the binding sites and then we detect the hydrogen bonds, that where the hydrogen bonds are possible. Because we are designing new lead compounds, so we will see that which of the lead compounds could possibly bind to these target proteins. So in target protein, binding site is identified and then different leads are searched against it to find out that which lead will bind better to my target protein. So that will be developed. And in de novo, we can look for these groups which are present in the binding site. So we can also design new drug molecule by simply designing those groups which can bind to these sites. So simply we know that double bond O can make hydrogen bond with this. Similarly here also double bond O can bind with this. OH group can bind with this double bond O. So now I know what functional group are required, so I can simply add the backbone to this. So this can also design new molecule, new candidate drug. So this comes under de novo drug development, in which I am starting from scratch. Then ligand receptor matching. So features must match chemical groups with similar properties. So this is known as ligand receptor matching. That means the ligand which I have considered, it should have those groups which can bind to my receptor binding site. So that particular matching is done, because if my lead is not having those groups which can make hydrogen bond and all those things, then it will not bind properly. So it will ultimately be filtered out by the criteria of binding affinity. So that thing come under ligand receptor matching, in which you match that the chemical group which are present in my target and what are the groups which are present in the lead. Either they are complementary to each other or they are not. So that identification is done here so that we can identify novel ligands. Pharmacophore concept, so it focuses on spatial arrangement of few key atoms and functional groups, and it used to search the database for molecular matching the spatial setup. So in many cases, if we know the target protein and we know the binding site functional groups, then we can simply search for those chemical compounds from my database which have the respective groups which can bind to this binding site groups. So I can shortlist those compounds. So that would be like targeted screening of different lead compounds. And this is the representation. So these are fingerprints, 2-point fingerprint, 3-point fingerprint and 4-point fingerprints. So which is telling you about different atoms and their properties, that how many cationic atoms are present, how many hydrophobic atoms are present, how many hydrogen bond donors are present, how many hydrogen bond acceptors are present. So when two such atoms are linked to one another, so that comes under 2-point fingerprint. When two different properties of atoms are linked to each other, and in this case only two properties are linked to one another. If there is link between 3 such functional groups, then it comes under 3-point fingerprint. And if there is linking between 4 different chemical groups, then it comes under 4-point fingerprint. So depending on how many such chemical or functional groups are present and they are connected to one another, it determines what will be the fingerprint. It will be 4-point, 3-point, 2-point. Okay, so this comes under pharmacophore models. So in pharmacophore, we are only focusing on the main groups which are responsible for the different interactions. So now the step is model development. So firstly, we select a training set of ligands in which we choose structurally diverse set of molecules, because ultimately we'll optimize them in the later sections. But firstly, we'll select different structurally diverse molecules, and these molecules will be used for developing pharmacophore model. Now for all these molecules, we will study their structures, we'll see what are the different functional groups which are present. So in this case, fingerprinting will be done, which we have seen here. So these fingerprints are identified for all these training set of compounds. Conformational analysis is done, which generates a set of low energy conformations, and these conformations are likely to include the bioactive conformations. Then molecular superimposition is done, so which comes under ligand based pharmacophore, in which I know the ligand from where I started. Then for all those ligands or all those training compounds which I have shortlisted, I will perform their superimposition to find out that in which positions do they vary from one another. So this is done for only ligands. So all those ligands are superimposed on each other, and then identify the similar or functional groups common to all molecules in the set. Then we find out that which of the functional groups are similar in all those chemical compounds. Next step is abstraction, in which we transform the superimposed molecule into the abstract representation, in which we will see that we will firstly perform average calculations, that which of the groups which are mostly conserved or mostly present in all those molecules. So those groups will be averaged, and that will be my abstract representation. For example, superimposed phenyl rings may be conceptualized as an aromatic ring pharmacophore element. Then validation is done. So a pharmacophore model hypothesizes the observed biological activity of molecule binding to a common biological target. So against one single target, we will look for the activity of all these chemical compounds, and this will be taken from chemical databases which already have this information available. So we will design this model which could be able to predict this particular biological activity by looking at the structure, and this model is valid as long as it accounts for the differences in biological activity across a range of molecules. And then we'll see that all these structural variations which are present in these ligands, they should be showing different activities. If they have similar activities, then that means structural variations are not bringing about any change in the chemical activity. So we select those pharmacophore hits which show sufficient change in the activity, or which vary in their biological activity. So this takes us to the top pharmacophore hits, and from these hits we can further move to our experimental validations and further steps for drug designing. Next is structure based drug discovery, which involves de novo drug design. So here we have seen ligand based drug discovery, in which we are starting with the ligand, but now in structure based drug discovery we'll start with the structure. So it involves using 3D protein structure information to develop new biological active molecules. So in this we will develop new drug molecules by taking help of already available target protein structure. Now key points are, in de novo, that means we are starting fresh or from beginning. So as I have told you earlier also, so in this case what we do is we look for the binding site of my target protein. We will see what are the functional groups which are present in this receptor or in this target protein, and then we identify all those chemical compounds which contain those groups which are complementary to the groups present in the target site or in the binding site of my target. So in this way, different leads will be identified which contain all those groups. Docking will be performed to find out which one of those lead molecules bind better, and ultimately shortlisting will be done. Different evaluations will be performed, and then ultimately potential drug molecule will be obtained after performing different in vivo, in vitro studies. So this is the overview of the de novo design, in which we are designing this particular drug molecule. So this was highlighted here. So in de novo design, because for the target protein we know the functional groups which are present in my target protein. So now I will be able to know that what will be the complementary groups which should be present in my ligand so that it can bind to the target binding site. So these are the respective groups which should be present in my ligand. So now what I will do, I will design a backbone which can contain these groups. So if that backbone will be developed, then it will give rise to new active compound which can bind to my protein or the target protein. So this comes under de novo drug design, in which I am designing drug from scratch. I do not have available chemical structure or available this ligand molecule. I am designing it based on the information of my binding site of the target protein. Then there are different steps. So first step is generation of potential primary constraints. So these molecules provide framework for desired structure with essential ligand receptor interactions. So whatever I have explained, these things are listed in points. So similarly in this, as I have mentioned, that you look for the binding interactions in the receptor, or you can say in the target protein, you look for the sites where it binds to the ligand. What are the groups which are present? So that way you are looking for the interactions. Now receptor based, that means interaction of receptors from the basis for the drug design, whereas it could also be ligand based, in which ligand could serve as key to the target. So similarly, if you know about the structure of the target protein, then you will see the groups which are present in the binding site. Or for example, if you do not have information of the receptor, then also you can go for de novo drug design, but in this case you will take help of the ligand. So if already ligand is available, then you will study its functional groups. So if you know its functional groups, then you can look for those functional groups. You will keep those groups present, and then you will attach the backbone by yourself to design new ligand molecules. So this also comes under de novo drug design. Then derivation of interaction sites. So this is crucial to model the binding site accurately, because you must know what are the interaction sites. And then it starts with an atomic resolution structure of the active site. So for example, this is my target protein, this is my binding site. So these are the amino acids which are present in my binding site. So now we can study these amino acids. So this will be my atomic study, in which I will see what are the atoms present in these amino acids. Either they are hydrogen bond acceptors, or they are hydrogen bond donors, or either there is a phenyl ring which is present. So all these groups are identified, and this is very crucial to design the de novo drug molecules. Now building up methods. So strategy is growing, linking, lattice based sampling, and molecular dynamic based methods. So now what you will do, you know all these groups, so you will design the complementary groups. So for example, if it is hydrogen bond donor, then you will keep the electronegative compound which can accept this electron. So for example, double bond O is required here. Similarly, for this benzene ring, this phenyl group, I need some compound which can show hydrophobic interactions. So that means in my other place there should be some hydrophobic group must be present. Similarly, in this case also, this is the hydrogen bond acceptor. So in this case the group must be OH, which contains hydrogen, so it can donate one electron. So in this way, if I have the information of the binding site groups, then I can design respective groups in my ligand, and then I can simply join those groups to one another. Now after linking these groups, linking is done. Now its activity will be identified, that whether it is active or not. And after activity is being calculated, I can grow also. I can also add other functional groups to optimize the structure of my ligand molecule. So after all this linking, lattice based sampling is done, then we need to perform molecular dynamic simulations, in which we will see that either this structure is stable or it can get disintegrated. So all those studies also need to be performed to optimize this particular structure. Then assay or scoring, so different interactions, evaluations will be done. Docking will be performed to find out the binding interactions with my target protein. Different search strategies could be used. Optimization, secondary target constraints could also be used to further refine it and optimize it. Yeah, so this is what comes under de novo drug design, and it depends that from where you are starting. So if you are starting with the target or receptor, then this is how you do it. And on the other hand, you can also do it with ligand based information, in which instead of looking for these groups, if you already know ligand, if you already have its functional groups, then same functional groups you will keep there, and you will add the backbone to it, you will link other groups to it. So that comes under ligand based de novo drug design. Okay, then comes docking based virtual screening. So in this case, when we say virtual screening, that means you have multiple compounds and you are screening them using computer. That is virtual screening. Docking based means if you are using docking method, that means you already have information of your target protein, you already have information about its binding site. So you are simply taking the chemical database and different compounds from it. You are using all those compounds and you are docking them one by one on your target protein binding site. And then you are calculating the binding affinities so that you can screen the compounds, all these million of compounds which you are using, and you can shortlist certain lead compounds which shows better binding affinities. So this comes under docking based virtual screening, which can reduce the number of chemical compounds with which you are working, and you can further proceed with the lead compounds. Yeah, now the next thing is that what are the molecules which are available for the screening. So in virtual screening, the molecules could be real molecules. That means 1 to 2 million in-house archives or 3 to 4 million commercially available. So from real molecules also you can take. Otherwise, you can also use hypothetical molecules, which means these are the virtual combinatorial libraries up to 10 raised to power 60 possibilities. So if you do not have already available molecules, then you can use hypothetical molecules. You can synthetically construct them. For example, in case of de novo drug design, as we know that we are constructing them, they are not already available. We only know about the functional groups which must be conserved, and then we are simply linking them to each other. So that comes under hypothetical molecules. So these molecules can be used for virtual screening. Then small molecule library categories, so journal libraries, which include the compounds which could be broad spectrum, which could be used for multiple disease or multiple target proteins, which are suitable for high throughput screening against various targets. Then comes focus libraries, in which we have designated ligands which are specific for particular target proteins, such as serine protease inhibitors, which specifically target the serine protease, which is enzyme. So which specifically targets serine protease, that means all the compounds which have one single target. So they comes under focus libraries. Then comes targeted libraries, which aimed at single and specific target. So for example, only HIV-1 protease, so all those ligands which only target HIV-1 protease, so if those libraries which only focus on one particular protein. Then comes cherry picked libraries, in which we have custom set of compounds which are synthesized upon request. So depending on, for example, if one particular disease is spreading a lot, such as COVID-19 as it spread, so that means in that time that interest of that particular protein increased. So we specifically picked that protein and identified the inhibitors to that. So that comes under cherry picked libraries, in which depending on request or demand, that particular set of compounds are studied and which are listed in these libraries, so we can use that information. So this is just the same thing. So query by target, in which this is the BRAF protein. So max selectivity score and max clinical phase approved. So depending on it, different structures are present. Now we will, based on that, which compounds we have to select. So selectivity compounds and clinical compounds, and then we will browse them. So most selective, approved, semi. This is just showing you that what are the different molecules which are available for screening and how we can select among those molecules, that which molecule we should select. For example, if you are studying this HIV protease or serine protease, then instead of using general libraries, we can simply go to focus libraries. So it depends what analysis you are performing. If you are specifically interested in one particular target protein, then you can go to focus libraries. Otherwise, if you are just doing exploratory search, then you can go to journal libraries. Then comes scoring functions. So we have seen that whenever we are doing structure-based drug discovery, in which we know the structure of target protein, then we are doing molecular docking studies. So in that case, we use different scoring functions to calculate the binding affinities and overall energy of my protein and protein ligand complex. So it involves scoring functions, which is an approximate mathematical method used to predict the strength of binding affinity between two molecules. So this is the mathematical way how we judge the binding affinities. So this scoring function can be classified into three main types. First one is force field analysis, second is empirical scoring function, and third one is knowledge based scoring function. So in force field analysis, this we have also seen when we were looking into molecular dynamic simulations. So this involves actual molecular forces, which will involve both known bonded and bonded interactions. So which will include the van der Waals, electrostatic forces, hydrophobicity to each force field, and this is extremely complicated, and this is the formula. So ultimately we are trying to find out what is the binding energy, what is the overall energy after binding between the ligand and the protein. So if this energy is high, that means structure is unstable and it might get dissociated. If this energy is low, then it tells you the structure is stable. That means the complex which is formed after binding of ligand and receptor, it is stable and it is a favorable complex. It will not dissociate. So that is what seen in force field analysis. We simply look for these interactions, that what are the value of these interactions. Are these interactions strong or these are weak? So if interactions are strong, that means the complex will be strong enough. And then empirical scoring function. So this is the fastest scoring function because it optimizes the empirical terms such as van der Waals interactions, hydrogen bonding, electrostatic energy, hydrophobicity, desolvation, and change in solvent accessible surface area. So all those parameters are taken into consideration while looking for empirical scoring function, and this is also fastest. Then third one was knowledge based scoring function, which uses experimentally determined structural geometrical data. So in this case we take information of the structure like PDB. So what is the distance? Because we have seen that if distance between two of my atoms, if they are close to each other, then there will be some interaction which will be present. And if two molecules or two atoms, if they are lying away from each other, for example, if this particular ligand, if it is away from the binding site, then that means the complex will not be strong. Whereas if it is close to if the distance between both of them is less, then that means the complex is strong. So this is what we identify in the knowledge based scoring function, in which we take help of the structural data or the geometrical data among both of them, and then we find that whether this will be a stable complex or whether the binding affinity will be high or it will be low. So these are the scoring functions to find out that which of the ligand is binding best to my receptor. Now the next concept is enzyme substrate binding. Okay, so now we have seen that this is how we figure out that which ligand has better binding affinities. Now there are different models which tells you about the different models which tells you about these bindings. So first model is lock and key model, and next is induced fit model. So in lock and key model, it assumes that my substrate, it is rigid, it is not allowed to move, and similarly my enzyme, which is my protein, this is also rigid. So this shows that there is rigid body docking among enzyme and substrate, so nothing will be allowed to move. That means all the conformations, they are fixed, there will be no change while binding or anything. Whereas in induced fit model, it tells you that as soon as the substrate binds to enzyme, it can bring about conformational changes. That means in this case, enzyme is flexible, or the protein is flexible, it is not rigid. And on the other hand, the substrate could also be flexible. So it depends which of the molecule you want to keep flexible. You can also keep both of them flexible. So in induced fit model, it assumes that during binding, there could be conformational changes in my protein molecule, or in my ligand molecule also, the conformational changes can be present. So these are different models and these are different hypotheses, because in docking, when we are looking for binding affinities, then we are also looking for the interactions. So during docking, you have to select whether you want to keep your protein rigid or whether you want to allow it to move. Next concept is one receptor and several ligands. So in that case, the concept of cooperative binding comes, because in previous case we were only talking about one ligand, but what if more than one ligand is binding to my target protein? So that in that case, the cooperative binding comes. That means when a protein binds to multiple ligands and the affinity for subsequent ligands changes after the first ligand binds, then it is known as cooperative binding. Now this cooperative binding could be positive also and negative also. That means as soon as my first substrate binds, it can also increase the binding affinity for the next substrate, and this can ultimately increase the binding affinity for all the substrates. Whereas it could be vice versa. That means as soon as my one substrate binds, it can reduce the activity when the second substrate binds. It might not allow the second substrate to bind properly, because it might bring about any change in the protein conformation such that second substrate could no longer bind to it. So that particular interaction is known as negative cooperativity. That means affinity is decreasing with the ligand binding. Positive cooperativity means affinity is increasing with ligand binding. Yeah, so these are the terms which tells you about different types of enzyme substrate bindings. Now predicting binding. So this is also the same, how you can predict. So first is lock and key model, which is easiest to compute because you are making your protein and substrate both rigid, so you do not have to make so many calculations, with minimal flexibility, and it involves 6 degrees of freedom, that is 3 rotations and 3 translations. Then if we talk about induced fit model, then it is quite complex, because now you are allowing your protein to move. So it requires modeling flexibility beyond 6 degrees of freedom, which includes movement of receptor side chains, backbone and the ligand. So in this case, you are making your protein also flexible and your ligand also flexible. So now more than 6 degrees of freedoms need to be calculated. So it will be complex, it will take a lot of time, although it will be better to represent, but it is time consuming. So it depends that what is your aim. So on the basis of that, you can select the model depending on your desired interest, because if you are selecting that particular enzyme which is quite flexible, which keep on changing conformation, then you need to make it flexible. You need to go for induced fit model. Then comes molecular dynamics and receptor flexibility. So this uses nanosecond long all atom MD simulations to capture protein conformational changes, and small molecules are docked to MD snapshot to account for flexibility. And next is MDIF protocol, which is designed to detect conformational changes which are not visible in short time scale, so which helps in opening or narrowing the binding site sub-pockets by atomistic simulations. So except lock and key model, all other models, they make the receptor molecule or the target protein flexible. That means the target protein is allowed to change its conformation, and in sometimes the conformation changes in very few time scale. So in that case, you can go for MDIF, which can even track for the short time scale movements or conformational changes. So this is how the binding is predicted. These are different models which are generally used. So this is the representation that how the data is collected. So from protein data bank you can get the structure of the protein, and you can study the mutations, affinity experiments, and crystal structure of the protein. So all these informations could be collected. MD simulations could be performed with the wild type protein or the mutant protein, and then ligand is taken. So ligand is docked into that particular protein, its affinity is calculated, and how its affinity is changing if you are making any mutation in the protein. So all those studies can be done, and then predictions can be made with respect to activity, how the different ligands or different mutant proteins, what is the activity, what is the binding affinity of the ligand with different mutated forms of the protein, and vice versa. So all those things, it is predicted. So this is also a type of drug discovery process only, in which you are designing or you are making changes in the target protein, or you can also mutate your ligand, you can make the changes. So ultimately you are predicting the activity versus the structure of the protein. Now how to select that which drug is the good candidate? So there are certain properties which need to be checked. So it should have good activity or selectivity on the right target, and then second thing is it elicit the desired biological response, but it should also be also follow the ADMET properties. That means it should be properly absorbed in the body, distribution, metabolism, excretion, toxicity. So based on the type of activity or what is desired, these properties might vary in the values. So there are different rules which have been given to find out that whether the drug is a good candidate or not. So all these things are checked. So now with the research we identify different drugs which could take 4 to 5 years, and we start with research, and then we develop the lead compounds, and ultimately those lead compounds which are developed, they are verified and they reach the market. So as you can see, these length of the arrows represent that how many compounds we start with and how many compounds we are left in the end. And in many cases in the development stage where the clinical trials are done, many cases the drug molecules, they have certain side effects, so due to which it does not reach the market and we fail. So then we need to perform further research, we need to further optimize them, and then development. So this thing keeps on going. So now the question arises that does the compound work in man? That means in vivo studies whether this compound is working, because in vitro study, for example, the compound is giving particular activity, but in vivo it is not giving. So failure rate is over 80 to 90%, and the reasons could be safety and efficacy. That means your compound is giving the activity, but it might not be safe, or it might not be effective, it might bind to some off target protein. So these are the reasons for failure. Now what can be done to solve this? So you can identify the ADMET problems, that where it is failing. Either it is failing due to absorption or due to excretion, metabolism, or either it is toxic. So all these things could be identified, and more emphasis could be done on these properties in lead optimization. So for example, if it has poor absorption, then we can optimize it, we can add those groups which can increase its absorption. So if we perform emphasis on all these criteria, then we can design better lead compounds which are less likely to get failed in the developmental stage. And these are the reasons of drug failure. So majorly the drug fails due to pharmacokinetics. That means everything is right, but as soon as they enter the body, they're not able to get properly dissociated or spread throughout the body. So that is one of the major reason of the drug failure, followed by this miscellaneous reasons, then lack of efficacy, animal toxicity. So these are some of the reasons which are responsible for the failure of drugs. And whenever we say ADMET properties, then this means whenever we take the drug, for example, different pharmacophore kinetic parameters are responsible for the action of the drug. So if we are taking it orally, then oral bioavailability means the fraction of drug dose that reaches the systemic blood circulation after first pass from liver metabolism. So as we take the drug, it will go to liver where it will get metabolized, and then how much concentration is reaching the systemic blood circulation is the one which tells you what is the bioavailability of your drug. So if bioavailability is poor, for example, if in metabolism only it is getting excreted out and it is not reaching the systemic blood, that means that drug is not suitable candidate. Then comes absorption. Absorption means how much portion of the drug dose that successfully crosses the gut wall. So in some cases the particular drug dose, it's not able to cross the gut wall, it get degraded. Then second thing is clearance. So volume of blood from which the drug is completely removed per unit time. So what is the clearance of that drug? How much time it takes to get cleared from the blood? If it is very small, then that means it will not have enough time to perform its action. Then volume of distribution, which indicates drug distribution throughout the body, that how quickly it distributes throughout the body. Either it is having poor distribution, and it depends that what is your aim. Either you want to distribute it throughout the body or it should be specific to one particular tissue. So this is how the volume of distribution is calculated, which is dose divided by initial plasma concentration. And this is the membrane permeation, that whether your drug will be able to penetrate or permeate through the membrane or it will not be able to permeate. So this is the calculation how the penetration rate is calculated. And now there are different factors such as half-life, oral bioavailability of a drug. So oral bioavailability tells you the dosing regimen, that is how much drug to be taken so that it becomes bioavailable. And half-life tells you how often you should take the drug. So if its half-life is small, that means after how much time it gets completely lost, or it's 50% of concentration is lost in how much time, so that you can design. There are certain drugs which we need to take after 3-hour intervals, then there are certain drugs which we take after 24-hour intervals. So it depends on its half-life and its bioavailability. So bioavailability will tell you how much dosage you need to take, and half-life, it decides that after how much time do we need to take that drug. So all those things need to be taken into consideration while designing the drug molecules. Then next thing is partition coefficient, which is the octanol to water model. So this tells you that whether your drug is hydrophobic or it is hydrophilic. So this will simply tell you that if your drug is hydrophobic, then it will not get dissolved in water. It will be only dissolved in the chemical compounds which are non-polar, such as in your membranes where the lipids are present. So it depends in which tissue you want to perform its action. So based on that, if you want your drug to reach the blood cells, then it must be hydrophilic. So this partition coefficient tells you that what is the whether your drug is hydrophobic or it is hydrophilic. So it's the ratio of concentration of compound in a mixture of two immiscible solvents at equilibrium. So equilibrium is the time when the concentration of the drug, the normal drug, is equals to the concentration of its dissociated form. So in pharmaceutical sciences, both the phases usually are solvents. Most commonly one of the solvent is water while the other one is hydrophobic, and for example it is mainly 1-octanol. So partition coefficient measures that how hydrophilic or hydrophobic a chemical compound is, and this is useful because hydrophobic drugs with high octanol to water partition coefficients, they are mainly distributed to hydrophobic areas such as lipid bilayers of the cells, whereas hydrophilic drugs which will have low octanol to water coefficient, they will be distributed throughout the aqueous region such as blood serum. So log D, it depends on the pH, and this is how the partition coefficient is calculated, and this is how distribution coefficient is calculated. So overall partition coefficient is simply checked to find out that whether your drug is hydrophobic or hydrophilic. So based on partition coefficient, you will be able to know that where you want your drug to which protein you want to target and where the protein is lying. So based on that, we can optimize our drug based on that. So we can make it either polar so that it becomes hydrophilic, and if you do not want it to be polar, then we can make other changes. So yeah. Then another thing is C log P, which is calculating the log P from the structure. So in previous slide, when we were looking for the log P values, the dissociation constant or the partition coefficient values, then these were the experimental values, whereas C log P, this is the predicted log P values by simply looking at the structure. So intestinal pH and drug properties. So these are if we talk about the intestinal regions, then different regions have different pH values, and we know that we have seen that log D, that is distribution, depends on the pH. So if pH changes, distribution also changes. So in this C log P, we can calculate for a particular chemical compound based on its structure that what will be the expected log P value, and what is the experimental log P and what is predicted, which is C log P. So this tells you that we will look for the structural properties. For example, we will look for the fragment. So there are 6 carbon molecules which are present. So its log P value turns out to be 0.78. So these are based on the calculations that already based on existing literature, what particular structure has what value. So on the basis of this, we can predict for different chemical compounds what could be the log P values, and we are not doing it experimentally so that it is reducing our time. So C log P is the predicted measure of compounds lipophilicity, which is calculated from fragment contribution, isolating carbons, and correction factors such as polarity and hydrogen bonding. So these things are used to calculate the C log P values, and it estimates log P based on structure. So in this way, this fasten up the calculation of log P because we are not doing it experimentally. So there is Lipinski's rule of 5 for intestinal absorption, which means that a compound, it should have hydrogen bond donors less than 5, hydrogen bond acceptors should be less than 10, molecular weight should be less than 500 daltons, and log P should be less than 5 for better intestinal absorption. So if some drug obeys all these rules, then that will be selected as the suitable drug candidate, and it will be ultimately released out in the market. And then there are certain examples such as the paclitaxel or the Taxol. So there were some rule violations in this particular drug. This violated 2 rules of Lipinski. First one was molecular weight. So its molecular weight was 837, which exceeds 500, and its hydrogen acceptors were 15, which is exceeding 10. Whereas its log P value was 4.49 and hydrogen donors were 3, which both were satisfied. But due to these 2 violations, there were certain side effects of this particular drug, such as hair loss, bone marrow suppressions, numbness, allergic reactions. So this is to state you that Lipinski's rule. So if some drug which follows all these rules, so it's most likely that that drug will be safe. But if it disobeys any of these laws, then it means it could have certain ill effects or side effects in the later future. So these were some side effects. Similarly, antimicrobial resistance. So antibiotics kills bacteria, but bacteria are protected by nature. So because there is some arm race which is going among bacteria and the antibiotics, so they try to adapt themselves so that they can survive. So this leads to constant battle between humans and nature, and it is never ending war due to nature's adaptability and resilience. So due to which it is projected that 10 billion deaths by 2050 could be there because of this antimicrobial resistance, because now so there are certain reasons for the development of antimicrobial resistance. So it is believed that many of the bacteria, they have adapted a lot, or many of the microbes, they have adapted in such a way that antibiotics no longer act on them. So this is what is assumed, that this could be the rate of deaths by 2050, and resistance rises as the new drugs are invented, but old antibiotics become ineffective. So as long as new drug is getting invented, now old antibiotics, they are becoming ineffective, and after 2011 there are very few new antibiotics being developed, and without antibiotics, diagnosed for death. Yeah, so this is what is shown, that how the antimicrobial resistance is develops. So due to DNA mutations, the bacteria has gained resistance, or due to horizontal gene transfer, due to lack of new antibiotics, due to overuse of antibiotics, or due to biofilm formation, and also the selective pressure which is acting, which is allowing them to adapt. So these are the reasons for antimicrobial resistance. So there are certain studies such as combating the antimicrobial resistance by targeting the enzyme beta-lactamase, applying therapeutic strategies. So because this beta-lactamase, which is synthesized by bacteria, so as we know that penicillin is the antibiotic, so penicillin contains beta-lactam ring, which interferes with the cross-linking of the wall of the bacteria, and which was leading to death of the bacteria. But now bacteria is synthesizing beta-lactamase, which is breaking down penicillin, so that bacteria is now surviving. So now we need to design some measures so that we can target this beta-lactamase. So therapeutics is to design new drug targeting intrinsic property of this enzyme. Enzyme need to be characterized and identifying newly emergent mutants of this particular enzyme, so that we can design more and more antagonists towards this enzyme. So hypothesis is that every beta lactamase are different with individualistic uniqueness. So that means this enzyme is not one fixed enzyme. There are certain modifications which are present across different microbial species. So first of all, we need to find those differences. We have to study each and every beta lactamase which is coming out from any organism, but this is impossible because thousands of genes, thousands of microbes which are present. So what can be done is to take help of interdisciplinary approach by combining microbial screening with computational and experimental techniques. So with experimental techniques, we can bring about the mutations in different sites by experimental means instead of isolating from different organisms. So this could be done. So samples could be collected, the genetic characterization could be done, the sequences could be identified, their mutants could be synthesized, and computational database could be made where we can store those mutants and protein sequences. And then we can clone them, we can overexpress them, we can purify them, and different characterizations and testings can be done in vitro, and then novel drugs could be designed with respect to that. So in the next slide we'll see. So there is anti-tuberculosis drug which is developed by inhibiting the beta lactamase with novel boronate transition state analogs, which is the BATSA. So in 2011 it was seen that 8.7 million people, they fell ill with TB, and new infections were occurring at the rate about 1 per second. So why this infection was occurring? Because the previously existing antibiotics were no longer working because there was resistance. And due to this beta lactamase of tuberculosis, it has been identified as major resistance factors. This was responsible, which was inactivating the antibiotics. So the reason how it works is that active penicillin, if beta lactamase is present, then it breaks down this particular beta lactam ring. It it opens up the beta lactam ring, due to which penicillin gets inactivated. And the beta lactamase has serine, which is in the active site, which is responsible for this particular breaking. So now novel inhibitors, which was BATSA, which was designed, the logic was that central boronate, it interacts with the catalytic serine OH. So now if we give BATSA, which is the cefotaxime, it already contains the boronate group. Now this boronate group, it will interact with this serine OH. So previously the active site serine OH of beta lactamase, it was interacting and it was opening up this beta lactam ring, but as soon as the boronate group is present, now serine OH will interact with this and it will pair up with this. So now in this case BATSA is present, then beta lactamase will bind to this BATSA and it will not be able to attack the penicillin. So now penicillin can work normally as it used to work previously, so it will act as antibiotic. So in this way novel inhibitors need to be developed so that they can target the beta lactamase. So method is to clone and express them and perform the purification of the compounds, and then different purifications are done. Kinetic characterizations need to be done by using different reporters, and then screening could be done of different compounds. So similarly this as boronate transition state analogs, so different analogs can be constructed, and for all of them calculations can be done which has better activity, which has better Ki values. So the goal is that Ki should be less than 10 micromolar. That means with this much concentration it should be more effective. So this is how what could be done to deal with the antibiotic resistance or antimicrobial resistance. And this is the example that different compounds were tested, out of which EC19 was the one which has the which is more effective with less concentration, with of 0.65 micromolar it was effective. And other than EC19, the another one was this PENM2, which is also showing the better results. So OD means absorption, that means the cell, how many number of cells are present, and this is the days. So this means with the dose, the concentration of the cells or the microbial colonies, they are dying off. That means this PENM2 and EC19, they are more effective than other drugs. And this is how the interactions are taking at the binding site of beta lactamase versus the EC19. So these are the groups which are involved in the interactions. And then there are certain other projects also which are related to beta lactamase inhibitor development, such as DBO moiety based novel drugs. So DBO is diazabicyclooctanes, which are class of serine beta lactamase inhibitors. So they are also the inhibitors which use a strained urea. So in previous case we were using the boronate ring, but now in this case we use the strained urea. So this particular group, this also interacts with serine, and this traps the beta lactamase enzyme so that beta lactamase could not open up the penicillin beta lactam ring, and now penicillin can better act on the microorganism. So example of this drug is NXL104, which irreversibly inhibit the beta lactamase from Mycobacterium tuberculosis. So this particular study was specifically done on beta lactamase obtained from Mycobacterium tuberculosis, and it was observed that as the concentration of this NXL104 increases, its activity was also increasing, and then this peak calculation was done. So this is to show you. So this A peak represents the free beta lactamase C enzyme, and these blue peaks tells you the beta lactamase enzyme complexed with the NXL104, which is my drug, you can say, which is the inhibitor which inhibits the beta lactamase. So this is showing that as there is increase in time from 0 minutes to 600 minutes, there is complex formation between the beta lactamase and NXL104 adduct. So in this case, this is the complex between beta lactamase and NXL. So as we move in time, the peak is kind of disappearing. So now there is no free beta lactamase. That means in this case, there is no free beta lactamase which could target my beta lactam or antibiotics. So this is to show you how effective this NXL104 is, because it is irreversibly inhibiting. It is not dissociating then. This is irreversible reaction. As soon as NXL binds, it do not allow the beta lactam C enzyme to dissociate from it, and these are the sites and interactions which are taking place with the serine. And this is the paper from where I have obtained these figures. So if you are interested, you can go through this paper, in which in detail they have mentioned that how this NXL104 is binding to beta lactamase. Yeah, so with this we are at the end of this topics. Now we will go through few MCQs. So now first question is, which of the following are the processes of traditional drug discovery? Option A is random screening, option B is trial and error method, option C is classical pharmacology, and option D is all of the above. Yes, so the right option is D. So these are all the methods which we have seen in traditional drug discovery. Now next question is, co-MFA method is used for option A 3D QSAR, option B 4D QSAR, option C 5D QSAR, and option D is 6D QSAR. Okay, let's see. So the right answer is option A, which is 3D QSAR. So three-dimensional QSAR is a computational modeling technique which relates the three-dimensional structural features of chemical compounds to their biological activity. So for example, in case of 2D QSAR, we are only looking at the molecular descriptors from 2D structures such as molecular weight and all those properties, but in 3D QSAR we take into consideration the rotations, the spatial arrangements. So there are different methods which are used in 3D QSAR, such as COMFA, COMSIA, HQSAR, GRIND, pharmacophore based 3D QSAR, and WALSERV, in which this particular co-MFA is comparative molecular field analysis, and this analyzes the steric and electrostatic fields using partial least square regression, and it is widely used because it is having good correlation with the bioactivity. Yeah, so the right answer was 3D QSAR. And if you are interested, then you can go into the detail of this method, that how it calculates the structure to activity relationships, and these are some other examples based on the type of QSAR model. Now next question is, which is not the physical property of compounds? Option A molecular weight, option B boiling point, option C solubility, and option D is acidity. Okay, you are saying option D. Yeah, so acidity is the chemical property, whereas rest 3 are the physical properties of compounds. Next question is atomic branching and ramification fall in which type of descriptor? Option A physicochemical, option B topological, option C quantum chemical, and option D is constitutional information. Okay, now let's see. So the right answer is option B, that is topological, because topological is associated with the structure. So atomic branching and ramification is generally comes under topological type of descriptor, whereas these are the example of constitutional. So this involves molecular weight, atom count and bond count, whereas physicochemical includes log P, solubility, boiling point, and in quantum chemical it involves HOMO-LUMO gap, dipole moment, partial charges. Yeah, so B is the right answer. Now next question is, what are the basic requirements for molecular docking? Option A protein 3-dimensional structure, option B library of small molecules, option C purified protein, and option D is A and B. Okay, so you are saying option D. Yeah, so the right answer is option D, because for molecular docking we require protein 3-dimensional structure as well as library of small molecules, so that we can perform docking among them and we can find the better binding affinities. There is no need of purified protein because these are in silico methods. So yeah, D option is right. Now next question is, in virtual screening, hypothetical molecules mean option A library of commercially available molecules, option B library of virtual combinatorial molecules, option C library of protein targets, and option D is library of FDA approved drugs. So what could be hypothetical molecule? Okay, so the right answer is option B, the library of virtual combinatorial molecules, because if we say option A, then this is the commercially available. That means these are real and purchasable. Whereas virtual combinatorial means you are virtually designing them. So these are designed in silico. So yeah, they are hypothetical. Whereas protein targets, so they are the receptor structures and these are not the molecules. Then a list of FDA approved drugs, so these are already existing. So that means these are not hypothetical. So yeah. Now next question is, what is the docking scenario between the receptor and ligand for the lock and key model in docking? Option A rigid receptor, rigid ligand, option B rigid receptor, flexible ligand, option C flexible receptor, rigid ligand, option D is flexible receptor and flexible ligand. So in case of lock and key model, okay, now let's see the right answer. So the right answer is rigid receptor and rigid ligand. So we have seen that in the lock and key model, it is assumed that both the ligand and my protein, they do not go any conformational changes, they are rigid. So that's why option A is right. Whereas in case of induced fit model, my ligand and receptor, they can be flexible and they both are not considered rigid at the same time. Okay, now let's move to our next question. So next question is, which of the following is present in Weber's rule of 5? Option A molecular weight less than 770, option B log P less than 9, option C is topological PSA is less than 140 angstrom square, and option D is all of the above. So the right answer is option C, which is TPSA less than 140 angstrom square. So these are the Weber's rule. So according to them, number of rotational bonds should be less than 10, TPSA should be less than 140 angstrom square, and sum of hydrogen bond donors and hydrogen bond acceptors should be less than 12. Whereas in this case, molecular weight less than 770. So this is not right, because according to Weber also, it also supports the Lipinski's molecular weight. So this is 770, so this should be 500. So that's why this is not true. Whereas log P, it should be, in this case, it should be less than 5. Whereas in this case, they are mentioning less than 9, which could also be 8. So this is also not true. So that's why option C is right. Next question is, which software is used for multiple alignment of sequences? Option A, Cluster Omega, option B, Autodoc, option C, Rosetta, and option D is FlexD. Okay, so for multiple alignment, we generally use Cluster Omega. Autodoc is used for the molecular docking, Rosetta is used for structure prediction, and FlexX is used for ligand docking. So the right answer is option A, Cluster Omega. Now next question is, which of the following is present in Lipinski's rule of 5? Option A, hydrogen bond donors should be greater than 5, hydrogen bond acceptors should be less than 10, molecular weight should be greater than 500 Daltons, and log P should be greater than 5. Okay, so you are confused among A or B. So the right answer is option B. That means hydrogen bond acceptors should be less than 10. Because hydrogen bond donors, they should also be less than 5. Whereas in this they are showing they should be greater. So that's why this is not right. Similarly, molecular weight should be less than 500 Daltons. Whereas in this they are showing it is greater. So that's why this is also not right. Similar for the log P also, it should also be less than 5. Whereas in this they have mentioned it to be greater than 5. Okay, so with this we are in the end of our MCQs. So now this is one of the review article which was published in Molecular Diversity in 2021. So if you are interested, you can go through this, because in this they have elaborated different stages in drug development, and they have also shown that how AI is helping in drug development and what are the things which are provided by AI which can now fasten up the process of drug development. Yeah, so if you are interested, you can go through this. And with this I am going to end the session, and this was our last session. So good luck for your exams, and if you have any doubts, then you can ask in the discussion forums, or you can also comment down in the video if you are seeing it in the YouTube. So if you have any doubts, then I'll answer, otherwise I'll end the session. And thank you very much. Okay, then I'll end the session.